start that.
Awesome. Thanks, Katrina. I am very excited to introduce Ramon for his talk today. Ramon is a research specialist and our CT manager here at the Museum of Zoology. He is from Toronto, Canada, and earned his undergraduate degree at the University of Toronto and his master's at the University of Calgary, both degrees focusing on vertebrate paleontology and biology. One recent and very cool accomplishment of Ramon's is that last year he actually published on the discovery of the oldest dinosaur fossils found to date from Alberta, Canada. Since starting at UMMZ in early 2019, Ramon has completed around 2,500 CT scans of museum specimens from across the vertebrate tree of life. And this has aided in data generation for a multitude of research projects. During the pandemic, Ramon has played an invaluable role in keeping our collections-based research projects moving forward. He's been scanning specimens, troubleshooting our finicky remote workstations, and holding down the fort at RMC, our Research Museum Center. Ramon is an excellent contributor to research projects and a wonderful collaborator, and we are grateful to have him at UMMZ. And with that, I will turn it over to Ramon for his talk. Please feel free to leave comments for Ramon in the Zoom chat, and we are going to open up the floor at the end of the talk to address those. Thank you.
Awesome. Thanks so much for that introduction, Taylor. And thank you everyone for coming to check out my talk. I'm really, really excited to talk about the work that we do at the UMMZ, and especially talk about 3D specimen visualization and CT scanning. This talk is going to sort of be a general overview of CT work in the context of zoology. I'm going to talk a little bit about what CT scanning is, some of the projects that we currently have going on, and a bunch of the different applications that we have for CT scanning. One thing that I do want to say is that there's a lot of visuals in this talk. So I'm going to have a lot of pictures on screen, similar to the one that you can see right now on the main title page here. And I won't necessarily be talking in depth about any one in particular, and some of them will have certain anatomy highlighted. If you do have any particular questions about these images, just let me know at the end, and I'd be happy to go back and take a closer look at them, because there are some really interesting animals sort of highlighted through here. Yeah, so with that, let's see. Am I moving to the next slide? Oh, there we go. All right, there we go. So with that, I want to talk a little bit about my background. So of course, I work for a zoology museum right now, but I'm actually trained, as Taylor mentioned, as a vertebrate paleontologist. And this is how I got my start with CT scanning. So during my graduate degree at the University of Calgary, I actually studied an extinct marine reptile called a plesiosaur. If any of you have seen these animals at a museum, you'll know that they actually have these extremely elongate necks. So I was actually interested in understanding how that particular structure functioned. So to try and answer questions about its functional morphology and biomechanics, I found a specimen, the one that you can see on screen at the top right, called Nicholsaura borealis, and we literally stuck it into a medical CT scanner, created a series of 3D models of its cervical vertebrae. And as you can see in my cartoon of a plesiosaur on the left-hand screen, we did a series of range of motion manipulations to look at how this particular structure may have function. So we looked at dorsal flexion, ventral flexion, as well as lateral motion of the neck to try and determine something about the ecology of this particular group of animals. So it was actually this work that I would say set me up really, really well for my current position, which is that of a research assistant and lab manager at the UMMZ, University of Michigan Museum of Zoology, where essentially my day-to-day job is running and operating our CT scanner. So this really means I work with a lot of specimens from our collections, and I'm scanning them constantly. I'm working with our students. I'm helping engaging in research projects and doing a lot of data management and, of course, 3D rendering, all things that I'm going to talk about and get into. But first, I want to talk about computed tomography, or CT scanning, and what exactly that is, because this is sort of the core concept of what I'm talking today. Essentially, CT scanning is an imaging technique whereby a series of X-ray radiographs are taken of an object from a number of angles around it, and then those radiographs are then taken and processed into something called into tomograms. And so tomograms are another set of images which represent cross-sectional slices of the object that was scanned. Originally, when this technology first came about, it was referred to as computer-assisted tomography, otherwise known as CAT scanning. So if you've ever heard the term CAT scan, CAT scanning and CT scanning are in fact the exact same thing. Now, without getting into sort of the nitty-gritty of the engineering of a CT scanner, which I cannot talk about as I'm not an engineer, I do want to point out a few of the features of a CT scanner, sort of the core components that allow us to collect data with these machines. So first and foremost, there is an X-ray source. So the X-ray source or emitter is where X-ray radiation will be generated and then fired from. There will be a platform of some kind where an object, specimen, or patient, in many cases, will be sitting. Then, of course, there will be a detector panel, and the detector panel is picking up the X-ray radiation or the X-ray beams that have traveled or attenuated through a particular or through your target. And so the detector panel, you can sort of think about it in the same way, or it's analogous really to the sensor inside of your digital camera, in your phone, in a DSLR, something along those lines. And then from the detector panel, we of course need image processing software and reconstruction software to make sense of the data that we're collecting. Now, generally speaking, there are two main configurations for a CT scanner. The first is one where the platform where the object, patient, or your target is sitting will remain stationary, and the X-ray source and detector panel, or in some cases series of detector panels, will rotate around that target. And this is what you'll most commonly see in the context of a medical CT scanner. Now, the other configuration that we have out there is one where the target platform actually rotates and the X-ray source and detector panel remains stationary. So in this case, it's actually the specimen moving as opposed to the radiation and detector panel. And this is what we generally find in micro CT scanners, which are the type of CT scanner usually employed in research and occasionally in industry as well. Now, for most people, you will have encountered a CT scanner in the context of healthcare or medicine. So CT scanners, as I mentioned, are configured for medicine, and they have been used in the healthcare world since about the 1970s. They offer a great range of utilities, in some cases greater than that of your regular old 2D X-ray radiograph, because it can allow healthcare professionals to look directly at the structures that they're interested in without having the overlays of other structures. So for example, if someone is interested in the rib cage, and particularly say your in your shoulder region, if they're using an X-ray radiograph, they will have to deal with the clavicle or the scapula, whereas with the CT scanner, they can look directly at the ribs. There are, of course, a wide range of medical uses. They can be used for assessing bone breaks, in cancer detection, in heart and brain assessments, and a whole variety of other things that I have not mentioned here. Now, if you have had a CT scan in the past and your doctor has shown you those scans, you might see something that looks like the images on screen right now. On the left, we have a series of tomograms, those cross-sectional images of a head. And on the right, we have, again, another set of tomograms of an abdomen of an individual. So in both cases, these are CT scans. And the main thing that I want to draw your attention to is the color gradient. So if you look at any one particular image in the set of images, you'll see that there is a black section, kind of a gray section, and then a white section. When a CT scan is conducted on an object, what you'll have is lower density regions of that scan showing up as black to gray, and higher density regions of that scan showing up in white or brighter colorations. In most cases, it's going to be white. So in the context of a biological sample or a patient, generally speaking, bone will show up as a bright white image, whereas soft tissues such as organs or musculature will show up in gray and be much harder to differentiate out from the scan. And of course, air will show up in black. And just really quickly, you can see these slices. These are the tomograms that I was referring to. So they are sequential cross-section through the target that was scanned. Now, for the rest of the talk, I'm going to be talking really about the scientific and research applications of CT, of which there are a great many. So as I mentioned at the beginning, of course, we use CT scanning in paleontology. It is a very useful tool for analyzing fossils, because when you take a CT scan of a fossil that may be encrusted in rock and matrix, the scanner can actually allow you to then differentiate between what's bone and what's rock, allowing paleontologists the opportunity to peer into fossil remains that could be hundreds of millions of years old. It's widely used in the geology world as well. So it can be used for contextualizing class sizes and mineralogical compositions in rock samples. It's also, in fact, used for studying meteorites. In anthropology and archaeology as well, it can be used. So last year, there was a study where they took mummies from Egypt. They put them in a CT scanner, and they were able to get all kinds of interesting anatomical information about these individuals that lived thousands and thousands of years ago. Actually, one really cool little study, I don't know, some of you might have seen it, but they scanned an individual and they were able to 3D print their voice box and then try and emulate the sound that that individual may have made using the CT data that they were able to gather. You can also use it for studying pottery and archaeological artifacts. Here you can see at the bottom right-hand screen, this is a Chimu whistling pot that we scanned for the Anthropological Archaeology Museum at the University of Michigan. And it's actually really cool because you can look inside these different objects to see what's going on inside of them. You can also 3D print them, which can be useful for educational purposes or display and study, of course. Now, something that people may not be aware of is how widely used CT scanning is in industry. So most of our mechanical household objects, so everything from like our cell phone to our kettle, at some point in its manufacturing process was probably CT scanned for quality control purposes. So CT scanning can allow manufacturers in the automotive industry, in the aircraft industry to look for things like microstructural flaws in the product. And so it's quite widely used to ensure that the products that we use on a day-to-day basis aren't just going to fail for all kinds of random reasons. So as you can see, there's quite a wide range of applications for CT scanning. Now, of course, we're talking about the Museum of Zoology, and CT scanning for us has really allowed a whole new dimension for us to be able to study the specimens that we collect. So here, for example, this is a specimen called Zonosaurus. It's a reptile from Madagascar. And this is it imaged in 3 different states. So the first is when the specimen was alive, before it was collected in the wild. At that point in time, researchers could have collected information about its habitat, diet, behavior, things like that. Then the specimen would have been collected. It's now preserved at the University of Michigan Museum of Zoology for future generations to use in their studies. You can look at things like its scales and whatnot just by looking at the specimen. But by CT scanning it, we can then look further, quite literally, into the specimen. We can look at its skeletal structure and morphology. But beyond just the skeletal structure from a single specimen, depending on the scanning modality that we choose to go with, we can get data on its soft tissue anatomy, neural anatomy, vascularization, reproduction, even things like parasite load. Of course, we can take these data and 3D print replicas of our specimens, which can be incredibly useful for education, outreach, and of course, research. So with all of the different potential data types that we can pull from a CT scan of a specimen, it really allows researchers, not just at our museum, but other museums and kind of all over, to ask quite a number of different questions about the specimens that we scan. So say, for example, you wanted to do an anatomical description of the morphology of a specimen. Say maybe it's a new species or a poorly known species. If we CT scan that specimen, we can extract out particular aspects of its anatomy. For example, in the case of this mammal on screen, we've gone through the data and processed out the nasal turbinate bones to characterize what those look like. Or in the case of the lizard shown on the right, we have pulled out the bones of the skull to look at the ornamentation pattern. And so you can take things like measurements of the size and shape of these different structures to better inform our understanding of the specimen, or just describe them in general if they're not known to science. But beyond just looking at the morphology, you can then take those data and contextualize them in a taxonomic, phylogenetic, and evolutionary context. So we can look at the different shapes and structures of the morphology that we pull from the CT scans and look at them across particular groups. So in this study, for example, they were looking at cranial morphology of a particular group of snakes, and in particular, they were looking at their fang morphology. Using CT data, they were able to gather quite a bit of information of the anatomy and the shape of the fangs, and then compare that across the group to teach, or to then write about, I guess, relationships across that group in evolution. But beyond evolution and beyond just shape, we can also study biomechanics and movement using CT data. In the past, researchers have taken the 3D models from a CT scan and put on, or using engineering software, actually work to understand muscle activation patterns. And this can be used to look at feeding mechanics, in this case, a species of tuatara, well, the tuatara from New Zealand. As well, people have been able to use CT data to look at joint articulation across the vertebrate tree to better understand how different animals have evolved their locomotor patterns. And in both of these cases, essentially what you're seeing is you're taking CT data and then pairing it with other forms of analytical techniques and imaging techniques to better understand how certain animals are essentially interacting with their environment and how they exist, behave, and what have you. Now, of course, with all of these different potential research questions that can be asked, having a CT scanner in-house at our museum means that we have an excellent resource to study the specimens that we have in our collections. But really quickly, before I go into our particular CT scanner, I kind of want to plug our museum. If any of you have not visited the Museum of Zoology at the University of Michigan's website, I encourage you to go over to our website. You'll see everything that we have going on in our collections. We have a massive collection. We have over 15 million specimens, and amongst our divisions, our individual collections are some of the largest in their respective fields. So, if you want to learn a little bit more about what we have going on at our museum, I really encourage you, go over to the museum's website and you can learn something about our collections. So back to the scanner. So as I mentioned, we do have a CT scanner at our lab, and this is an excellent tool for our researchers. But a little bit of history about it. It is a Nikon XTH225 ST micro CT scanner, which is a mouthful, and none of you will ever need to remember that name ever again. It was purchased as a part of a facilities upgrade for the Biological Sciences building over on the main campus at the University of Michigan. However, it was decided to house it at the Research Museum Center, which is where the UMMZ is located. And this has turned out to actually be an excellent decision because we are in seemingly a unique case where our specimens are about 50 feet from our scanner. And this is excellent because if we want to scan something, we just roll the cart, walk for about 30 seconds, get the things that we need to get, bring them over to the scanner, and away we go. So it's a really, really great situation to be in. So the scanner came down to the RMC in July of 2018, and it's been functional since August of 2018. I started in January of 2019. As Taylor mentioned, we've been scanning pretty much nonstop since the beginning. Since August of 2018, we have conducted about 3,100 scans, which represents about 2,600 specimens. So some specimens are actually scanned multiple times, depending on the research project and depending on if we're looking at particular regions of their anatomy. Now, pre-COVID, we were scanning about 12 to 15 specimens per day with 1 or 2 additional overnight scans. So the different types of scans, so during the day, we generally would run a skeleton or sets of skeletal scans on specimens where we're just looking at their bony anatomy. And in the evenings, we would be conducting a different type of scan called a DiceCT scan, which I'm going to get into a little bit later. Another thing that's going on is we are working to set ourselves up to become a core scanning facility. Now, generally speaking, I just want to go back to this image here. In the middle, you can see kind of a handle on the door. That's the cabinet in which we can scan specimens. Generally speaking, in a single scan, we can scan specimens up to about 30 centimeters in length from head to the end of their tail. That's to get the whole specimen in one scan. However, we are able to scan larger specimens. It just takes a little bit more work and a little bit more planning. So actually last summer, Dr. Cody Thompson, our mammals collections manager, was able to get a red fox, and we did scan it. So vulpes as shown on screen. But in order to do that, we had to do a series of scans. So we would put the entire specimen into the scanner and then scan the head and pectoral region as shown in the image labeled scan A. And then we would do a second scan of essentially the main body cavity of the specimen, and then a third scan just of the tail and kind of the hind legs. Now, what we're then able to do is take those 3 data sets and then stitch them back together to get one larger data set representing this large specimen that we otherwise would be unable to scan in a single instance. So it does take a bit of work, but we are able to scan some large things. I've even scanned a fairly large old world vulture in the past as well. Now, on the flip side to scanning large things, we're also able to scan some very, very small things. The smallest specimen that I've had go through the scanner to get a good scan of was a 5 millimeter long dog tick. And while large specimens present their challenges, small specimens also present their challenges. Now, when we go to scan something in our scanner, we want to always ensure that it's stabilized so it doesn't move during the scanning process. And often this involves packing foam and packing peanuts directly up against the specimen. Now, in the case of things like an insect like the wasp shown on screen, they're very fragile and have all kinds of little projections on them that we don't want to damage by stabilizing the specimen or accidentally damage when we pack it. So generally, we'll try to put these specimens on a pin. And then what that actually causes is quite a bit of movement. However, it does take a bit of time and you can get a good scan. And when you do, you can get some very interesting results, such as the one shown on screen, where we can literally go through and pull apart different segments of the body of these specimens. So there's quite a lot of utility and ability to scan specimens both large and small and everything kind of in between. Now, as I mentioned, we do another type of scanning, which is called DiceCT scanning. And DiceCT stands for diffusible iodine contrast enhanced scanning. And essentially what this scanning modality does is allow us the ability to pick up soft tissues on specimens. So as I mentioned at the start of my talk, when you scan a specimen or target, whatever, the more dense an object is, the whiter it will appear. And this allows us to then differentiate those structures. With DiceCT scanning, essentially what we're doing is taking more radio opaque or less dense structures and artificially making them more dense so that they can be picked up in our CT scan. And to do this, essentially we use an iodine solution called Lugol's and stain the specimen for a period of time and then scan them. And as you can see here on the right, this is a blarina or a shrew. And if you look at it in the top right-hand quadrant image, that large kind of round structure towards its bottom is its heart, as well as around it, you can see all of the musculature. Now, normally, if we were just to take this specimen and put it into the scanner, we would only see the bone. So with this staining process, we're able to see much, much more anatomy than we normally would. And this is actually a very powerful tool, especially in the context of a zoology museum. So for us being museums people, we're concerned with the long-term use and care of our specimens. And one thing that DiceCT essentially allows us to do is a set of non-destructive digital dissections on our specimens. So by staining the specimen, if we, for example, wanted to now see some of its internal anatomy, we could quite easily do that by digitally dissecting away the anatomy that we're not interested in, such as in the case of this particular specimen, which is a snake that we DiceCT scanned in the past. Here you can now see its brain with other parts of the anatomy not shown, as we're not interested in them. And so we can literally go through the specimen and pull out the particular parts of the anatomy that we're interested in. And then this specimen is still in the collections and can still be used again for future research projects. Now, quick comparison between these two scanning types. A skeletal scan, generally speaking, is a much faster scan. It takes about 15 minutes. We can pack the specimen, stabilize it in that way that I mentioned earlier, get it in the scanner, get the scan completed, reconstruct the data, and then put the specimen back into the collections. For a DiceCT scan, it is quite a bit more involved and requires quite a bit more prep time because the staining takes about 7 to 30 days, depending on the size of the specimen. It is a passive process, so we essentially put the specimens into the stain and allow the iodine to slowly bind to the specimen. The scan then takes about 3.5 hours. The reason the scan takes longer is because we need to change some of the settings on our CT scanner to try and pick up the finer structures associated with soft tissue. So things like muscle and nerves, these are all much more fine and still, even though they're stained, quite a bit less dense than bone. So we need to change some of our exposure settings on our CT scanner to be able to pick these up. But the really time-intensive part of a DiceCT scan is actually the destain period. So, of course, once we've stained the specimen and collected our data, we're really happy, but we want those specimens to return to the collections in a good condition so that they can be used for future projects that we can't perhaps conceive of at this time. And so we destain the specimens in alcohol. And again, this is a passive process, at least as we use it at the University of Michigan, to slowly leach out the stain. Now, this is just a primer for DiceCT. You could probably do an entire talk about DiceCT alone. So if any of you have more in-depth questions about this process and what we do, I'd be happy to answer those or send me an email at some point in the future if you are interested in doing this work yourself. Now, regardless of whether we're doing a DiceCT scan or a skeletal scan, our general workflow in our lab is the same. The specimen gets packed. It is then placed in the CT scanner. And like I said, in our scanner, the specimen rotates relative to a stationary X-ray source and detector panel. The immediate output from the scan is a series of radiographs. So these images look the same as the type of image that you would get from a 2D radiograph at your doctor's office. It's then that stack of images that gets reconstructed into the tomogram. So if you can see on the top right-hand corner, that series of cross-sectional images through the specimen. Again, you'll see the bone in bright white, and then the softer structures, actually in this case, it's just the bag around the specimen shown in gray. From there, we can take those data and actually create our 3D model. So I've been showing a few images of different 3D renders of our specimens. This step called segmentation is actually how we go about getting that. So essentially, we will take our 2D cross-sectional images, load them into a segmentation program. So there's a number of these programs available for people to use. There's a Viso, Volume Graphics, Dragonfly, Mimics, and 3D Slicer, and several others. Some of them are very expensive and some of them are free. Critically, you need a lot of computing power to be able to actually do this. If you have a gaming computer with a powerful graphics processing unit, generally speaking, you'll be fine to do this sort of work. And as I've mentioned before, the 3D renders generated from segmentation can be used for a variety of different research projects. It is a time-intensive step, though. You do spend a lot of time looking at your cross sections, slowly drawing or tracing over them to highlight the structures that you're interested in. However, once you go through that process and actually do this segmentation, you are able to highlight and see all kinds of anatomical structures. So in this case, you're seeing on screen an image of, actually this is a silky anteater that's been segmented. In this case, we pulled out the mandible, the cranium, the atlas axis, as well as sort of the central claw that it uses for climbing, and I think for some aspects of its feeding as well. So although it is time intensive and computing power intensive, the results are really, really cool. So of course, with all of these tools and resources that we have on hand at the UMMZ, we do have a number of ongoing research projects. And I primarily work with 3 PIs and then on 1 NSF-funded project that's going on. And I just kind of want to quickly show off some of the work that they're doing without going into too much depth. So with Dr. Dan Roboski, we're working to scan a whole set of skinks. So these are lizards that have variable limb loss. And there's a student who is going to be looking at their pectoral and pelvic girdles to sort of characterize the anatomical sort of features that go along with these differential limb losses across this group. Then with Dr. Allison Davis Roboski, we're characterizing the venom delivery system across a group of snakes. Here you can see on screen, that's actually the venom gland. In purple, the venom delivery duct and then fang of a snake. I don't recall what species it is right now. But essentially we're going through and doing a massive, massive DiceCT project where we have probably at this point just over 100 snakes scanned and being processed by everyone in her lab. And then with Dr. Hernan Lopez Fernandez, his lab, they're really interested in a wide variety of questions regarding morphology and taxonomy of various fishes from mostly South America, but from some other places as well. And then of course there's the Obert project, and that's a whole big project that I've dedicated a few slides to that we're going to talk about in a few moments. Now, as you can imagine, we've scanned a lot of specimens, and this results in a lot of data being produced. A single data set can be anywhere from 10 to 30 gigabytes once we include all of the raw data, the reconstructed data, and any derivatives, so 3D models and what have you. So in addition to storing everything on hard drives, which isn't necessarily a good long-term solution for data curation, we are working with 2 data repositories to make our data both available and archived. The first is the Deep Blue data repository, which is hosted from the University of Michigan's library. So you can go there and Google, or sorry, not Google, you can search by specimen number, and you can find some of the data sets that we have currently uploaded. And we're still working to populate that repository as of right now. And the other big one is, of course, Morphesource. Morphesource is an excellent resource for finding 3D data, especially CT data. And we have hundreds, if not over several thousand data sets up on Morphesource right now. And if you were to go to Morphesource, what you would find are 3D meshes. So say you wanted to do 3D printing or just some 3D work, you could go there, find 3D meshes such as the snakes that you can see at the bottom right-hand corner. And of course, image stacks of tomograms. So those tomograms that can then be used in your own segmentation projects or whatever you have going on, you can pull data from there and you can use it, in your research projects, you can use it for educational purposes, what have you. Actually, one thing that's really cool with Morphesource is a new version was just rolled out, and they have a 3D viewer tool that's available. So even if you don't have a very powerful computer, you can actually go there now and take a quick look at some of the data in 3D. It'll render it there for you. It might be a little slow depending on the machine that you're working on, but it does work and it's pretty interesting. So to go back to Obert, sorry. This is the other very large project that I'm a part of, as well as a number of other people at the UMMZ. So Obert is an NSF-funded project, which is part of NSF's Advancing Digitization of Biodiversity Collections program. essentially what the Obert project is looking to do is increase the visibility and accessibility of museums' collections in the United States by using CT scanning. We've started to use photogrammetry for a few collections, but overwhelmingly, the goal of this project is to CT scan a large amount of fluid preserved specimens in institutions within the United States. I believe the goal is about 20,000 specimens, as you can see there. This would represent about 80% of extant genre. And we would also be doing, and so that's just skeletal scans, we would also be doing soft tissue DiceCT scans of about 60% of extant families. And so again, all of this data will be made available online at Morphesource for people to access and download and use for their own research projects. As I said, this is a multi-institutional project. It's actually headed out of the University of Florida by Dr. Dave Blackburn. And then these are the other institutions funded through the project, 16 of which are actually museums. There are only 6, including the University of Michigan, that are imaging centers. So for all of the other collections, they will be sending their specimens to the imaging centers to get scanned and then have posted on Morphesource and all the other interesting things. Now, of course, wanting to scan over 20,000 specimens can be a bit of a logistical nightmare. And so as a big part of our project, I just wanted to recognize how we kind of go about organizing ourselves. So we've used iDigBio, which is a data aggregator that pulls together information about specimens and collections from across different institutions. And essentially, we've come up with a series of hit lists. So there's a mammals hit list, a herpetology hit list, birds hit list, and fish hit list. And essentially, like you're seeing on screen right now, it shows which institutions have the specimens and then where those specimens can be directed to for scanning, and then who has scanned them and when they've been scanned if they're uploaded. So it's a lot of spreadsheeting and a lot of organization to try and get the scanning done that needs to be done for a part of this project. And as of January 2020, we've actually scanned about 7,700 fluid preserved specimens from about 42 US institutions. So we are actually pulling specimens from institutions outside of just those that are funded. And although 7,700 may not seem like a lot of the 20,000 goal, when you break it down, you'll see it's actually quite a big accomplishment in that, like, almost 90% of all amphibians have been scanned, 60% of reptiles, 30% of fishes, 25% of mammals, and 10% of birds. So we do have more work to do, but even just right now, as we speak, there's over 5,000 specimens available for download on Morphesource. So of course, this is a little out of date. It's about a year old. We haven't done our annual report for the year. So the numbers are actually larger than those shown here. And I know I've mentioned a couple times, anyone can go on Morphesource and download these data for their own research. However, interestingly, we are getting a lot of downloads for educational purposes too. And not just educational purposes at a university level, but also at a K through 12 level. So we're getting a lot of teachers who want to introduce their students to science and anatomy, getting on Morphesource, downloading the data, and then using them in their classroom. So really what this project in a way is allowing to happen is to literally bring museums and their collections into the classroom. And so this is a very powerful, potentially a very powerful tool for engaging students and children in science at an early age. And of course, there are other people have downloaded the data for 3D printing, art, and other various kind of outreach projects as well. Now, kind of before I get to the close of my talk, I do want to kind of touch on what we've been doing during COVID-19. Of course, as you hopefully have kind of gathered from my talk so far, CT scanning and working in this lab is a very hands-on project. And like everyone else, we have been locked down for a lot of the last year. And so we have had to try and find ways to rejig our work plan and ensure that things still get done. Of course, for a period of time, no scanning could be conducted in our collections. However, we could still do things like data processing. We could still render 3D models, and we could still do a lot of data uploads to make data available for people to access. So just before we went into lockdown, we kind of worked, myself and several of the PIs and other people in the museum worked to help set up remote workstations for our students and research staff so that they could continue to do their research projects and access data. And this, again, was another whole logistical undertaking, because each of these remote workstations, of which we only had 4, could handle 1 user at a time. So I had to go through and create a number of calendars and a number of instruction manuals so that people, essentially could continue their work during COVID-19. And of course, for me, once everything was set up, I was no longer scanning. So I had to find, in a way, new things to do. And I went through the data and I was creating 3D renders of specimens that we had. I was creating tutorials for our students and staff, and of course, helping to continue support research and troubleshoot things as they came up. Now, if any of you follow me on Twitter or have seen my Twitter feed, you might recall that from about April to June of last year, I was doing a daily segmentation. And this kind of happened by accident. I was doing these 3D renders and I thought people might like seeing them. And so I tweeted out a couple and I tweeted some information with them, and I found that people were really engaging with them. So during the course of COVID-19, I found myself doing far more outreach than I had done before by posting images like the one you can see online, which is just a very little bit about the anatomy of the specimen and a little bit of information about its ecology, as well as some resources for people to learn more about these animals. In some cases, these were also animations. And these animations are all posted now on the university's or the UMMZ's YouTube page. So if you want to see some stuff, you can go on to YouTube. You can look up the University of Michigan Museum of Zoology, and there's probably about 20 of these animations available. Another thing that we're working towards setting up is a Sketchfab account and kind of page for our specimens. Sketchfab, if you've never been to it, is sort of a 3D model social media page, which allows people to hop on and click around and engage directly with 3D models. In some cases, there are annotations that can teach you about the anatomy of these different specimens and allow us to provide you with more information about them as well. So we're kind of working on getting that live as well. As I mentioned, created a series of segmentation tutorials. So if there are any of you who are interested in learning how to work with these data, we actually, myself and one of our undergrads, created a whole kind of web series that walks you through what CT data is, how you can use it, what programs you need, and essentially everything that you would need to get started working with this data. It's also on the University of Michigan's, the UMMZ's YouTube page as well. So going forward, I have been back scanning in the lab since about July or August of 2020. However, because of all the precautions we have to take due to COVID-19, this is at a much reduced capacity. So I'm generally scanning about 6 to 8 specimens per day, and doing about the same number of DiceCT scans since those run overnight. I'm continuing to support research, engage with students, and do some public outreach. I don't tweet as many of these 3D models right now just because I've been in the lab, but I do the occasional one. And finally, one of the long-term goals of that we have going forward is setting up our CT lab to become a core scanning facility, which would allow the CT scanner to become a resource for the wider University of Michigan and EEB research community to hopefully do their own projects and get really cool data like the kind you can see here on screen. So with that, I just have a few acknowledgements. Of course, the University of Florida, University of Michigan, Obert, NSF, Dan Roboski, Allison, Hernan, and everybody else named on this screen. And so with that, thank you. And I'll take questions, I think.
Thank you so much, Ramon. Yeah. All right, so if anyone has any questions, give you some time to put those in the chat, or you can also ask them out loud.
Are there chat questions?
Not yet.
All right. Oh, what kind of voxel resolution are we getting? So the finest resolution I can get is about 5 microns, but it takes a bit of work to get a specimen scanned at that resolution. Most of my scans happen between 10 and 120 microns. The sort of coarsest we can get is 121.62 microns. Can we use CT scan on dried plant specimens? I do believe we can use it on dried plant specimens. I haven't tried yet. I know that paleobotany people have scanned, leaf impressions and things like that. So it is possible. I have not tried it yet. Can we scan? Let's see. Is Cody's fox the largest thing we've scanned? I tried to scan a camel skull the other day that was probably bigger than the fox. It didn't work. It was too big. So I guess the fox is probably the largest thing I have successfully scanned so far. So this is actually a good question. What is the downside to leaving a specimen stained with iodine? That's actually a really, really interesting question, because DiceCT is a relatively newer technique. I would say people have only really been doing it for about 10 years now, and it's only really been perfected en masse in the last 5-6 years. With, of course, fluid preserved specimens, we're always worried about desiccation, and the staining process that we currently employ takes the specimens out of alcohol and puts them into water for staining. And so there can, of course, be some desiccation to the specimen. It looks like they generally return back to good shape, like they kind of plump back up once they go through the destaining process. In terms of color, the coloration will change for the stain, but we can take most of that stain out. However, I will say long-term negative effects of DiceCT staining are not fully known as of right now on our wet preserved specimens. All right. What's been the most surprising finding while scanning? Oh, I wish I'd actually put a picture up of it. There was a snake. I guess Allison's group collected. We hadn't looked at it that closely. We scanned it, and in its gut was an entire frog. So we actually have a scan of a snake and then a whole scan of a frog that's inside of its gut as well. Try a coconut. I guess we could, yeah, Brad, if you want to bring a coconut to the lab, by all means, we'll stick it in. What would be my #1 species to scan if I could scan it? I don't know, actually. We've scanned so many cool specimens. I remember being really excited when we scanned the Sphenodon. Yeah, we got to, I don't know, there's all kinds of things. I'm just excited in general for scanning in a bottle of liquid. Okay, so yeah, I guess I didn't sort of explain that really well, but so for specimens that are wet preserved, so the question is, how do you scan a specimen in a bottle of liquid? Do you have to take it out and dry it out or something like that? So we will take the specimen out of the alcohol, but we won't dry it out because that will have negative effects on the specimen. Generally, what I will do is pack the specimen into plastics and kind of like a heat sealed bag. And then we'll pack that into a plastic size appropriate container and then put foam around the specimen or packing peanuts to stabilize it. And then once the scan is done, the specimen can go back into alcohol. Is the size of the scanner the explanation for the bottleneck? So is the size of the scanner the explanation for the bottleneck in the scan size of the mammals? Yes. Yeah. So if we had a bigger scanner, we could scan larger specimens. Actually, so we're being in Southeast Michigan. Interestingly, I have heard that Ford and Chevy have CT scanners that you can drive a whole vehicle into, and they will scan, to look at every different part of their vehicles or their competitors' vehicles probably as well. All right. So museum specimens have become increasingly important sources of DNA, particularly for rare, endangered, and extinct species. Is there information on how CT scanning may damage DNA and what, oh, sorry, I kind of, and what we can do to increase specimen utility while minimizing damaging biomolecules. That's a really good question. I haven't seen any published work on the potential damage towards DNA on specimens, but I know a big thing right now in the field of, I guess, zoology in general is trying to get good DNA from formalin preserved specimens, because I do know that formalin can have negative effects on specimens. So I'm not sure exactly what negative effects CT scanning is also adding to the mix, but depending on the history of a particular specimen, how it was prepared, how it was collected, there could already be degradation. But that's actually a really good question. I think someone should be looking into that. Is there a plan to scan our placental elephant? I don't know. We should ask Cody. Is there? Because we have one, Cody. We should stick that in there. We did scan placental quadruplets of an armadillo, and that is available on Morphesource. We actually also did a DiceCT scan. Because of their stage of development, the bony structures were not that well resolved in the skeletal scan, but when we did the DiceCT scan, we could actually see quite a bit more structures on them. Oh yeah, glad you liked it, Gail. Is it possible to scan spiders, and how would you prepare this material? It should be possible to scan a spider. From what I have seen, actually, interestingly, with insects, it seems that you can get a lot more information from just a very short 15-minute scan of an insect than you can of a vertebrate. There's a lot more harder structures inside, and I've actually seen musculature come out of insects just from a non-DiceCT, what would be equivalent to like a skeletal scan. How we would prepare for this, yeah, it would be interesting. We'd have to find ways to sort of pin up the different appendages so that they don't move, because we wouldn't want to pack those specimens into kind of a bag and put foams and packing peanuts around them, as I'd be worried that would crush the various arms and things hanging off of them. How many times? Last question, it looks like, how many times do you expend to scan a mammal skull? I think you mean like how long does it take to scan a mammal skull? It can take about 15 minutes, generally speaking. Problems with fossils. Yeah, so fossils, they offer a whole nother set of challenges, generally speaking, because fossils are much more dense. They are a longer scan, sometimes at higher powers. You can use different filters to reduce some of the attenuation effects that the X-rays will have when they travel through various minerals that might be in the matrix around fossils. But we actually don't scan too many fossils on our particular scanner because the paleontologists have their own machine. They actually have the exact same scanner themselves. Yeah.
All right. If those are all the questions, yeah, everyone's saying good talk and thank you so much. Oh, wait, sorry, one more came in.
Oh, right. Last question. So do we have a supercomputer in the UMMZ dedicated? We don't have a supercomputer, but we do have a set of very, very powerful, essentially gaming computers. They've got like 128 gigs of RAM, and these massive dedicated graphics cards and everything like that. So yeah.
Cool. All right. Well, thank you so much again for talking with us today.
It was a lot of fun.
Thanks everyone for coming.
Thank you all.
Yeah, and we'll see you next week at our seminar. Same time, same place.